All right, we're going to be covering exoenzymes now. There's a number of different types of plates. First plate up is the starch agar plate. The enzyme we're looking for here, just like we saw with the other biochemical tests that we were looking at, the enzyme we're looking for is amylase, and what it does is it hydrolyzes starch, breaks it down into short polysaccharides, and then into maltose ultimately. The indicator in here is iodine. So basically we just add regular old grams iodine to the plate after it's been incubated for a while, and anywhere that we see a clear zone, that means that amylase has acted upon the starch and removed all the starch from that area. You can kind of see it here in this picture, but Staphylococcus epidermidis is negative for amylase. Bacillus subtilis creates this clear zone, and it's got a nice little clear area, so that's amylase positive. Here's a little better picture of it. You can see it's got this nice, real clear zone around it. It's not super big, but sometimes it will get bigger depending on how long you leave the plates incubate for. This is only a 24-hour incubation, so you can see it's only separated a little bit. Key thing with adding iodine to this plate is you only want to add just enough to barely cover the plate. If you flood it, then it's not going to show up quite as well. So just add enough to cover the surface and let it absorb in. Otherwise, you won't see anything very clearly. The next one is a spirit blue agar plate. This has the enzyme lipase that we're looking for. It breaks down a number of lipids. The indicator in here is a lipid called tributyrin. Add it to the plate. It's special because it is heat labile, so it'll break down in the autoclave. We have to actually add it in after we make all the media and let it cool down to an appropriate temperature, otherwise it'll break apart. Now, the trick with this media is clear zones, non-greasy areas, are an indicator that it is positive. Color change is not technically significant. So the story behind this media is a long time ago we used to think that the color changes were significant, except that in certain cases some bacteria showed up as a false positive. They cause a color change and you go, oh yeah, that's positive. In reality it's not. What you're basically going to do is, instead of having a hazy, greasy layer, you'll see this little clear zone occur around the bacteria. Here's a slightly better picture. You can see lipase positive has that little clear area where the bacteria basically eat up all the lipids and clear up the plate, so it's no longer greasy. If you guys look at the plate when you guys initially get it, you can see that it's kind of got a waxy, oily layer on the surface, and that's just kind of the way the plates normally look. So if you see the waxy, greasy look go away, then you can say, oh yes, that's definitely lipase positive. The next one is milk agar plate. And we're looking for basically proteases, anything that hydrolyzes any kind of protein. In here, the indicator we're using is casein, which is the major protein of milk. We're looking for a clear zone, basically where the milk has been eaten away by the bacterial proteases, and there is no longer milk there. So here you can see a great example of a protease positive bacteria. It's destroyed all the casein here, and now the milk is no longer opaque here, so we can see right through it. This bacteria, of course, doesn't have active proteases, and so it doesn't eat up the milk around it on the plate. Gelatin tubes are basically looking for the enzyme gelatinase. They hydrolyze gelatin, so they basically break it apart. The indicator here is refrigeration. So after we inoculate these and put them in the incubator, at incubator temperatures, they will still be liquid. But if we put them in the refrigerator, if there's gelatin present, it will gel up and basically be a semi-solid. However, if it is gelatinase positive, it will remain a liquid because all the gelatin's been degraded and it will no longer firm up. So if it's liquidy, it'll be positive after refrigeration. That's how you differentiate them. The last test is litmus milk. Now, litmus milk is a little bit confusing, especially if you look at some of the results online or even in your microbiology atlas. But basically, we can cover a number of different factors in here, but we're only going to be looking at a couple of them. Basically, we're looking for the ability to ferment, produce acids from lactose, and also the production of enzymes, proteases. It basically causes the proteolysis of casein. The indicator in here is litmus, which is a pH indicator. I'm sure you guys have heard of the word litmus test before. It normally starts out as this kind of light blue, purpley colored broth. When you inoculate it and it's positive for lactic acid fermentation, lactose fermentation, so lactic acid is the byproduct, and protease positive, it will create a nice clear pink coloration. However, if you inoculate it and it creates a curd, because the proteins will start to unravel because of the acids produced from lactose fermentation, and the bacteria doesn't have proteases, it will form a white curd that kind of sits at the bottom, and then a little pink layer where all the liquid remains on top. Really key here, though, is to make sure that you don't disturb it. So when you pick it up, don't shake it too much because you'll make it kind of very difficult to read the results. So just keep it very still when you take it out of the incubator so that you don't accidentally cause a jostling of it. There's a lot of other results for this as well. Maybe very, very different depending on the bacteria. We don't need to get onto all of them, but we're just going to show you a good selection of them here. So just keep that in mind. Here's another example of those in case you guys were interested. You're going to see just a wide variety. So this is the control. This is how it normally looks. If it's just got the lactic acid, the acid and lactose, it's going to form the nice pink layer, and it's got protease, so it doesn't have that curd. If it's got proteases and the ability to create alkaline amino acids, it will change it up to a kind of lighter purple coloration. So that means it's an alkaline scenario. If it just has proteolysis, that is, it does not ferment at all, this will stay this kind of dark brown color with a little bit of the protein sitting down here at the bottom. So it basically just turns this dark color. If it produces an acid but casein remains, then it will stay down here at the bottom. And this one has a light amount of acid but not very much, so it shifts the pH just a little bit. So usually weak fermenters cause this to happen. Not necessarily really important to know, but just something that you should be aware of, that there's multiple ways of reading these tubes.